30 days left for Re-NEET. And if I ask you the order of enzymes in DNA replication, most students mix up which enzyme acts first and which enzyme acts only on the lagging strand. That one confusion is why students lose 4 marks. Because NEET does not test mugging up, it tests whether you understand why each enzyme comes. By the end of this video, you will know which enzyme acts first, why primase is needed at all, which enzyme works only on the lagging strand, and the one direction rule that explains the entire chapter. Most students memorize DNA replication as a random list of enzymes. That's the mistake. The order is not random. Each enzyme exists to solve a problem created by the previous step. So don't memorize a list, understand the chain. The first enzyme of DNA replication is DNA helicase. Helicase breaks the hydrogen bonds, which are non-covalent bonds, between base pairs and opens the DNA double helix. This creates the Y-shaped replication fork, but opening the helix creates tension ahead of the fork. The DNA starts supercoiling. So another enzyme comes, topoisomerase, also known as DNA gyrase in prokaryotes. Its job is to remove supercoiling tension ahead of helicase. Now another problem appears. The separated strands want to join back again. So single strand binding protein SSB protein attach to the strands and keep them separated. But now comes the most important question. Why do we even need primase? Because DNA polymerase cannot start synthesis from nothing. It cannot initiate DNA synthesis de novo. It needs a free 3' OH end to begin adding nucleotides. So primase synthesizes a short RNA primer around 10 nucleotides long. This RNA primer provides the free 3' OH end needed by DNA polymerase. Primase does not make DNA. Primase makes RNA. Now DNA polymerase III attaches and starts building the new strand. And this is the most important rule in DNA replication. DNA polymerase synthesizes DNA only in 5' to 3' direction. Always, no exceptions. This one rule creates the entire leading strand and lagging strand concept. Think carefully. If DNA polymerase works only in 5' to 3' direction, what happens on the opposite strand? That strand cannot be synthesized continuously. So it is formed in small fragments called Okazaki fragments. One strand is synthesized continuously towards the replication fork, that is the leading strand. The other strand is synthesized discontinuously in fragments away from the fork, which is the lagging strand. And because fragments are formed separately, the lagging strand needs multiple RNA primers. Now, the lagging strand becomes messy as RNA primers are present between DNA fragments. So two enzymes comes for cleanup. First, DNA polymerase I. DNA polymerase I removes the RNA primer and replaces it with DNA nucleotides. So remember, RNA polymerase III is the main builder and RNA polymerase I has the janitor role. Without polymerase I, RNA would remain stuck inside the DNA strand. But even after primer removal, small gaps called nicks are still present between fragments. So DNA ligase comes and seals those nicks by forming phosphodiester bonds. So the full causal chain is: helicase opens the DNA, gyrase removes the supercoiling tension, primase lays the RNA primer, DNA polymerase III builds the strand, and DNA polymerase I removes RNA primer and replaces it with DNA. And lastly, ligase seals the gaps. Every enzyme solves the problem created by the previous step. That is the real memorization trick. Now open NCERT class 12. Chapter 6. Read this line carefully. DNA polymerase catalyze polymerization only in one direction, that is 5' to 3'. This exact line creates NEET PYQs every single year, and the trap option is always 3' to 5'. In NEET 2022, question came: DNA polymerase catalyze polymerization only in one direction. That is, now most students mark 3' to 5' because they remember the template strand is read in 3' to 5' direction. But NEET is asking about synthesis direction, and DNA synthesis always occur in 5' to 3' direction. So the correct answer is 5' to 3'. Another favorite NEET question: which enzyme joins Okazaki fragments. Now, most students often mark DNA polymerase because they think the enzyme that builds also joins, which is wrong. The correct answer is DNA ligase. Polymerase builds and DNA ligase seals. And remember, ligase acts on the lagging strand because Okazaki fragments are present there. One last step students get confused about is DNA polymerase I and DNA polymerase III. These are two different enzymes. Polymerase III builds the main strand while polymerase I removes RNA primer and replaces it with DNA. So if NEET asks you which enzyme removes RNA primer, the answer is DNA polymerase I. Now a quick recap. 3 things you must remember. 1. The causal chain. Helicase opens, primase primes, polymerase III builds, polymerase I cleans, and ligase seals. Number 2. DNA synthesis always occurs in 5' to 3' direction. And number 3. Ligase joins Okazaki fragments on the lagging strand. Different enzymes, different jobs. In the next video, we will cover the cyclin trap, the one NEET students keep getting wrong. See you in the next one.